So welcome everyone. We have Joanna Holbrook from BenevolentAI, Fiona Neilson from Repositive, yeah, and we've got Hugh here also from Kheiron Medical, which is great. Thank you very much for coming, guys. You're going to get very bored of my voice here because I'm here for another 2 sessions, so I do apologize. But let's roll into the next session, which I'm very, very excited about because a lot of you people have heard in the news a great deal of stuff recently about breakthroughs in liquid biopsies, the fact that AI is now outperforming radiologists and histopathologists. Those are the guys that look at kind of slides when you have a biopsy. And a great deal is happening in real practical application of AI, deep learning, machine learning techniques to actually make cancer something that we much more deeply understand. And part of it has also triggered some pretty big investments, not only in the private sector, but also in the public sector. Only a couple of weeks ago, since the sad passing of Baroness Tessa Jowell, the UK government has initiated the Dame Tessa Jowell Brain Cancer Mission to try and really understand and actually use adaptive molecular-based treatments to attack currently what is an incurable disease. But not to just do that in a tiny little trial, but to do that at a national scale. But to do that, we need technology that is literally only just emerging, and it's stuff that some of you people in the room and some of the people on stage here are actually developing. So what I'll ask the panel to do, apart from Stefan, is to just very, very quickly introduce yourselves, tell us what you're passionate about, what you're working on right now, and then we'll roll into some questions.
Shall I go first?
Hugh, go first, yeah.
So I'm Dr. Harvey. I was a consultant radiologist at Guy's and St Thomas's Hospital, but I got very bored and frustrated in the NHS at the slow pace of uptaking technology. I would sit in front of a computer, which in 2016 still used Windows XP. I then went to go and work at a small company called Babylon Health for a year, and then I'm now working at this company called Kheiron Medical, where we are 100% focused on finding breast cancer faster and more accurately than ever before.
Fiona.
Great. Yeah, hi, I'm Fiona. I'm the founder and CEO of Repositive. My background is in computer science and then in bioinformatics. I used to work in analyzing cancer data, and I got really frustrated when I realized that although I'm a believer in precision medicine, I realized that precision medicine will not become a reality if we don't unlock the necessity for data to input into all the research that's going on in preclinical research, because if you don't have access to the data, you don't get any results. So I realized this when I was working at Illumina. You can generate data very quickly, you can use the fastest and greatest algorithms, but if you don't have access to the data you need, you don't get very far. So that's why I started Repositive, and now working to enable all the researchers with the data they need.
So if ever you want to find a mouse that matches your cancer, you go to Fiona right there. Excellent. Joanna.
Hi, I'm Jo. I'm a biologist, and I've been working in drug discovery. I worked in targeted therapies in Big Pharma, where we started talking about personalized medicine and matching drugs to 1 or 2 somatic mutations. We know it's more complicated than that. There's a huge amount of information out there, as Stefan mentioned, in the biomedical literature, in the kind of deep, high-dimensional data that Fiona's collecting, and yet it's not getting to clinic. And that's partly because we need the AI to find the vulnerabilities in the cells and to find these smart combinations, but we also need the drugs, and we need to get the drugs faster and better. So I'm working at BenevolentAI, where we're using AI to speed up the drug discovery process.
It's a very interesting span of companies here. So we've got people that are working at the very, very beginnings of how you find molecules that will fit the problem. Then we've got in kind of Fiona's field, it's the move from discovering those and how quickly we can turn them into candidates ready to put into humans. And then Hugh on the end there is actually working in the field of, okay, then how do we get all these new things and including new technologies, not just new molecules, into the hands of our average oncologist, our average doctor? And well, Stefan's just here to throw a spanner in the works, frankly. So let's start basically on Jo, on your side. But I'd really like to make this kind of interactive if possible. Tell us about what you're seeing really in, I mean, how long does it take today for typically, without the normal processes, without the new processes, without new AI, how long and how expensive is it to get 10% more lifespan out of out of somebody with cancer? So it's really extending out the question, not how much does it cost to build a drug, but how much does it cost to actually get us to something that gives us just an extra 10%, let alone 100%?
That's the problem. And the industry stated standard is 10 years to develop a drug from first idea, first observation to something that can be used in clinic. And the problem is that when it gets there, you're asking the patient to take a big chance, because the drugs that we develop them to very imprecise diagnostic codes or to one somatic mutation, so EGFR positive cancer. And so the majority of patients that fit that group may not respond to the drug. And you're asking that patient who doesn't want to waste their time, doesn't want to take the potential side effects for a drug that doesn't work for them, to take that kind of risk. I thought what Stefan was saying about shots on goal, most people get 2 shots. And so what we need to disrupt is not just how fast we get to clinic, but also allowing us to find the patients for the right drug combination, so people get the drugs they need precisely for them. And that means we have to be less simplistic about characterizing, say, a tumor. We need to use all the data that places like Genia, like Illumina have created at patient level, so we can be smart about picking those combo drugs.
One of the big questions that I always get asked about all the drug discovery companies, though, is don't we already have quite a lot of drugs that hit a lot of targets? Why is it that we're just trying to find new drugs for single targets? We've kind of been left with systems biology problems, not simple problems, and they usually are very, very difficult to crack with 1 or 2 bullets. You need to find 20 or 30 needles in a haystack that are all moving at the same time. So how are you approaching that at Benevolent?
So we're trying to learn from patient data to select those particular combinations. And as you say, some of those needles have already been developed. Some of those drugs are there and in clinic and improved, but not for the right indication. So it's finding the network biology problem, the pathways that are perturbed in that individual tumor or in individual cells in that tumor, and finding the right combinations of drugs. And we can do that learning from pre-existing data on combo efficacy and network biology of those particular cells.
I'm just going to bring it over to Hugh here. You were a practicing radiologist. What if I told you that you didn't just have to report on a little lump that you saw on a CT scan, but I described that lump in a multi-dimensional description that only an AI could understand, and even though you would definitely stick a needle in that to biopsy it, the computer says, nope, there's only a 0.0001% reason for you to do that, and it would be bad. How would you respond, Doctor?
I mean, that's super exciting, really, really exciting. The less that we actually need to do, the less invasive procedures, the fewer times a patient has to come into hospital, etc., the better. And what you're describing there is where we are heading with AI, deep learning techniques, looking at image perception tasks. We're at the very nascent stage now of applying these technologies to radiology, where we can find big macroscopic features. We can tell you if there's a lump in your breast, or we can measure the size of something that we see in your CT scan. But what you're talking about is being able to look at functional imaging, underlying imaging biomarkers. Radiomics is the name of the field. And I think it's a really exciting area to be in. We have a long way to go. I'm not saying that this stuff is here and we're ready to treat everyone and scan everyone with this stuff. But I think we do need to really consider putting investment, funding, research, time and effort into combining not only the work we do with drug discovery, but if you're treating a tumor with a new drug, you need to follow it up with radiology. You need to image it at regular intervals to see if it's getting bigger or smaller, or what the function of that tumor is doing underneath. With functional MRI, we can now find the the diffusion, the blood flow through tumors. We can see if that's changing. We can see choline levels. We can see all sorts of chemical biomarkers. We can see on mass spectrometry, MRI. So if we could start applying AI and deep learning to these technologies, can we do very simple, quick, fast imaging techniques to see how tumors are changing with treatments? And that's an exciting area. Sorry, I have blathered a bit, but it excites me. So
not at all. I want to ask Fiona a question as well, because a few people who are very well resourced and knowledgeable and networked, they very often, even as individuals, let alone as big sort of R&D companies, they try to grow the cancer inside them in mice. It's called xenografting. Terribly exciting, and I'm not sure, it's very interesting, the ethics around this is how many lives of how many mice is worth one human, but you literally grow the cancer in the mice and see what grows and then test lots and lots of drugs on those mice. And you can also do that in cell lines as well. How effective is that? Is that something that we're really learning from? Or is that just a shot in the dark and seeing what sticks?
Yeah, so just to give a little bit of history or background, just for the people in the audience. So these kind of mouse models were first used for exactly that purpose of treating a cancer patient. You have these 5 drugs to choose from. Which one should you choose? If you choose the wrong one, you might either kill the patient or you might not have any effect, which means that the cancer would progress so much that it would be too late to have a second choice. So the idea of the mouse models in the first instance, when they first started using these, was to use them as an avatar for the patient. So you take a bit of the patient's tumor, put it under the skin of a mouse, and grow the tumor in the mouse, and then try with different drugs to see which of these drugs are actually effective on the live tumor. So it's as an avatar for the real. Instead of putting drugs in the real patients, you try it on the mouse first because there's a faster turnaround, and then you can try different drugs, and then the drug that works, you give it back to the patient. Now, this is, since this was first invented, and it's a great invention in the sense of a way of testing new drugs directly to improve the patient care of the individual. Like, this is really personalized treatment we're talking about. So since then, it has become a whole industry, because this means that this is actually a way for biopharmaceutical companies to test drugs in mouse models before these drugs go into clinical trials, which means that you have a much better chance of identifying which particular subtype of tumor will this drug actually be effective in before you start putting it out to a larger cohort of people where you know that this is more likely to be effective because you've already seen it effective in mice. Of course, there's not a direct one-to-one correlation about from what is effective in mice to what is effective in humans, but there's a much bigger chance that if this drug actually makes a tumor shrink in a mouse, then there's a bigger chance that it will actually be effective in the human. So this is a very it's been an important discovery. It's been, I'd say, exploding in use over the last 5 to 10 years since this mechanism was first developed, this use of mouse models. And now it's so commonplace that it's now an FDA requirement for any drug before it goes into clinical trial. You have to first show that it actually has an effect in either a mouse model or an in vivo or an ex vivo model where it's based on an actual patient tumor tissue that you show a drug is effective before you start putting it into real patients in a trial. So yes, it's a very very specific way of showing that the drugs are effective, and you can use it to narrow down this particular set of biomarkers, characterize the tumors for which this drug is effective.
But we're still talking about biology here. We're still talking about the time it takes for a cell to become 2 cells, to become 4 cells, to react to something, in which time I'm already on my, I'm already on my 4th or 5th cycle of chemotherapy. My cancer's decided, I don't really like this cocktail I'm drinking. I'm going to change, and so on and so forth. So actually, I'm going to ask Stefan this question. In your personal experience of that kind of space, the limitations of biology, what more can we do in silico for the real patient that is live with, with a course of treatment currently underway?
Yeah, so in my case, we worked with a company called Cellworks. They're a Sequoia-funded investment, and they do pathway modeling of tumor cells. So what does that mean? It means that you look at your mutations, you do sequencing of the tumor, you look at what mutations does that tumor have. So how are the mechanics, internal mechanics of the cells affected by that? And there's what in these cells are what called these pathways of how molecules interact with each other. And what they tried to do is predict how a combination of drugs would affect these pathways, right? And what you saw is that if you put in an individual drug, then the cell would react to that and it would find a way around that, right? But what they were able to do in the model is to say, well, these 5 or 6 drugs in combination, they can actually, if you put them all in together, they can target the cancer in a way that it doesn't have an escape pathway. Now, the interesting thing about this is that most drugs that get approved today, they need to show efficacy, and they need to show efficacy in sort of randomized control trials where you don't do anything else. And a lot of these drugs fail in these sort of stage 3 trials where they can't really show efficacy very well. Patients don't improve at all or only improve a little bit. It doesn't mean the drug doesn't work. It just means it doesn't work as a monotherapy, right? And it's very hard to design these combination trials because the combination that you need is different for every patient. So you'll never get enough patients to show it with statistical significance. So a lot of drugs that actually do have the potential to affect the cancer don't make it through approval. And in a model like this where you have the software to help you guide it, you could get use out of that drug. In fact, one of the Trump candidates for the new head of FDA was a guy that Peter Thiel recommended, who came to sort of a bit of fame because he was a sort of gay VC speaking at the Republican convention the first time as an openly gay person and supporter of Trump, and he was not very well liked in Silicon Valley. But what he did was he put forward a candidate as a new head of FDA who said, it's not the FDA's job to test for efficacy, okay? It's safety only, right? As long as the drug is safe, that's what the FDA should check. Efficacy, let the market decide, and who knows what's effective, right? It may not be effective in monotherapy, but it might be effective for some patients in combination therapy, right? Which would basically allow a lot more drugs to come through and also bring the cost of drug development down. By the way, that got shot down, not so much, you would think that the pharma industry supports that, but the pharma industry actually likes the current regime because it kind of keeps them at the top of the food chain. It makes it harder for sort of startups to come up from the bottom. So they were violently opposed to that.
I told you you'd put a spanner in the works.
To run trials with that kind of power to show efficacy.
It's interesting. So let's talk a little bit about regulatory now, because Stefan's brought it up. There is absolutely no question that we are moving forward with more discovery. I would sooner today, and this is really very much a hand-on-heart admission, I would sooner today trust computer scientists and data scientists to tell me how to treat a really complex systems biology disease like cancer than I would my fellow oncologists. That does not necessarily mean that the role is not there. There are lots of subtle things about life and choices and what you expect of the years that you've got ahead that clinicians are very good with. But that for me is a huge change. I would not have said that 2 to 3 years ago, that I would trust a data scientist, a group of AI people to help me navigate through my choices in cancer. But the regulatory bodies think very, very differently to that. And in fact, most of the people that are given the authority to write that prescription for you, which might be the better prescription than the standard of care, it might be far better, also do not accept that. They don't trust that. What are we doing to overcome those Berlin walls?
I've sat in quite lengthy meetings with the MHRA, who are the regulatory body for the UK, and they follow the regulations set out by the European Commission, the Medical Device Directives for Europe. They have no clue how to regulate AI, machine learning, deep learning technologies. They don't have the staff who understand these techniques. They don't know what metrics they should be assessing them by. They don't have the manpower to even start looking at all of the possible applications. I am slightly probably more cautious than you and Stefan. I think we do need some element of regulation, otherwise it's going to be the complete Wild West and everyone's going to be selling their new AI-powered drug willy-nilly. I think we do need some kind of regulation, But these guys, the MHRA, need help in this country to do it. And Brexit is going to put a massive barrier in front of that if it happens. I'm quite impressed by the FDA's approach. I don't think they've gone quite far enough yet. They've just down-regulated, only last month, in radiology, any AI system from class 3 down to class 2. Class 3 is the kind of device that can kill you, you know, and these things clearly don't kill people. So they've only just got there, so there's a long way to go.
Seems obvious to me there's so much data out there. Every working scientist feels constantly guilty because there's no way you can read all the literature in your own discipline, never mind everything that you should be reading that's contextual to that will allow you to make the breakthroughs. And that must be the same for every clinical oncologist. You can't possibly read all of the new advances coming out. We're going to need machine learning to help us make sense of the huge amount of data that's there, that's human readable in the form of journal articles and the way these things are, but also the kind of high-dimensional data that Fiona's making available to everyone. It's not going to be possible for any human mind, even a very clever MD, to imbibe all of that information and find the best combination for this particular person. So it's obvious that we're going to need AI to do this. How we build the trust, I guess we have to do that by data too. We have to show that it works. And it might even be that we're doing retrospective studies in existing data.
But your clients, a lot of them are going to be the very organizations who are not encumbered by how quickly things happen. They are actually the organizations that can live long enough to be able to wait for the next blockbuster or, you know, work through incremental change, which doesn't help the patient that has 2 years to live. And so what are you seeing from the big biopharma companies that, to Stefan's point, are definitely not lobbying against change? Let's face it, we have to be realistic here. There is absolutely no question that 10 years and several billion to get a blockbuster drug and then living on that in revenues for another 10 years and hoping that someone doesn't come up with something through AI in 6 months after that is the bedrock of the industry. What is pharma doing about this? Is it disrupting itself? Is it really going for it? Is it really embracing it?
So my clients are patients. Benevolent's a biotech. We're trying to get drugs to market. Pharma, I think, are worried about being outcompeted by people who are going to get there faster, and I think that's where the incentive will come from. It's clear that you can get 100% response if you are careful enough about selecting the patient population, and that will outcompete the blockbuster drugs, which are coming out slower and slower. Maybe there's not much low-hanging fruit left, and pharma's productivity is going down. So I think that there is the incentive because of the need to compete.
And if we look from like a high-level perspective, what's happening in biopharma these days is that they've already made very effective everything that's in the clinical way of the pathway to getting a drug to market on clinical trials and so on. It can still be made more effective, but right now there's a lot of the efficiency gain because they're all interested in shortening this time. I mean, the individual researcher is not interested in spending 10 years to get this drug approved. They want to do it fast and they want to get real results that are reliable. And this efficiency gain now, it's evident that it comes in all the preclinical work, because if there's systemic changes made of enabling easy access to data, for one thing, using the right tools, making the collaboration and communication work across the industry, that these different, both the biopharma companies, the big guys can move further, faster to get to the market, but there's also this, what do you call it, the democratization of the initial work can happen in many smaller individual activities that are then picked up in the bigger pipelines and fed into the market via the big biopharma. And this is a trend you're seeing. I mean, all of the, if you look at where all the drug discovery and new targets are developed today, it's all happening in the small companies that are coming up. And then once they get a drug validated enough that it looks like it's a candidate for the clinic, then it's picked up by the big biopharma that buys it out and takes it to market. And you'll see more and more of this.
Stefan.
Yeah, I mean, the one thing to sort of understand here is that the big pharma business model is essentially at threat of being broken by this development, right? So traditionally, these were these small molecules, it costs about $1 billion to bring one to market. You have to run these trials with thousands of patients. Keep in mind that these trials are run as profit centers by the academic institutions. So you pay between $10,000, $20,000, sometimes $30,000 a patient, like even at Stanford, right? They say, well, which trial, they join a few trials a year, right? And it's baked into their revenue stream, right? So patients get put on the trials that they happen to be running there, right? As patients, you have to make some difficult choices, right? So they would tell me, well, if you do this, then you won't qualify for that trial. So in the end, what I had to do was essentially find a doctor who was willing to keep completely separate medical records so that Stanford thinks I'm this patient, I'm a vanilla patient, and in the real world, I'm a completely different patient. Right?
That's Chatham House rule. Don't tell anyone.
But basically, if you look at the drugs that are being developed, right, for example, the drugs we developed, a lot of these are now short peptides, basically short protein chains. So you use software, you sequence the tumor, you use software tools to figure out what that peptide should look like. It's effectively an antigen that you can produce. So 12, 20 amino acids long. Any lab in the world can produce that. The software spits out a drug that's specific to me, right? That's never going to get sold again to any other patient in the world. And even if it does, if it is an antibody for a mutation that multiple patients have, even if you do have IP on it, it's so simple to produce. Same way you can 3D print something at home. Any lab with minimal infrastructure can produce these peptides, or in some cases you use viruses with some payload on them, to sort of genetically modify the DNA cells. And again, modifying a virus to put that piece of information in. Simple technology. So small labs are going to do that. And even if you do have IP on it, it's going to be completely impossible to enforce. So it's true democratization of these drugs, right? They are going to be developed by the AI, by the software. They're going to be printed and made somewhere the same way I found a manufacturer. And the US guy said it'll cost you $1 million to do it. And I found a German guy to do it for $40,000. And they say, well, it's not GMP, not good manufacturing practices. It's still a great lab, right? But it basically doesn't meet the FDA standards. So I said, well, I'll go with the $40,000 version of this, not the $1 million version, right? For my 12 vaccines.
It's really interesting. So as a clinician, I'm going, whoa. And as a geek, I'm going, wow. And it's going to be very, very interesting to see who it is that's going to be on the advisory boards of the future to the FDA and the regulators. Is it going to be more people who are going whoa, or more people that are going wow? And I really look forward to seeing that change. There's something though that still I wanted to ask you guys, and I ask this as a clinician, what are you doing to help educate the likes of me and my colleagues towards some of this stuff? Because I'm going from having learned a bunch of stuff at medical school, qualified 20 years ago. That stuff is basically an iteration on things that have started about 2,500 years ago and, moved on. And every generation we see a bit of a change here and there. And we've done, there's quite a few iterations after 2,500 years of things change in a glacial pace. But now things are going to change monthly. I mean, I'm not even saying yearly, but we're talking about monthly, or even going from the choice that everyone should be making to a choice for that particular patient at this time. This is fundamentally different. What are you guys doing to help people like me?
So Professor Eric Topol has come over on the invitation of our Secretary of State for Health to do a review of the impact of technology on the NHS, and he's invited me to co-chair the AI panel. So we are actually looking at how we educate an entire workforce, a new generation of not just doctors, but all of the allied healthcare professionals. And one of the things I'm really pushing for is that we have a new class of profession within healthcare, which is the algorithmists, people who understand this technology.
Algorithmologists.
Algorithmologists, whatever you want to call them. And because I believe that algorithms are the new drugs in a way. It's this technology which is so complex and difficult to understand. We need a whole new sort of profession in medicine. So I'm working as part of a panel with Health Education England to see how we can possibly even begin to educate an entire workforce on that. But not just the workforce, the patients as well, because it's them who are the ultimate deciders of their own care.
Where do I sign up? I thought it would be a really cool thing just to get the questions out from the audience before wrapping up, if that's okay, because I know you're dying to say your final pieces, but we will at the end. Has anyone got any questions to ask these folk? Aha, there is Nenad over there with his hand up. I think that's Nenad?
Yes.
It is.
So, Mike, so Stefan, the normal therapy that you have you broken new ground to the point where you can actually publish the stuff that other people that have your cancer can then also benefit from it?
I'll repeat the question. Yeah. So regarding Stefan himself, have we reached a point where we're able to publish some of this work where other people will be able to benefit?
Well, have there been enough new breakthroughs from the things you've been doing?
I'm sure there will be publications coming out of that. For example, out of Stanford, the academic group that we developed the monitoring test with. That's a first. That's never been done in this tumor type. It's very difficult to monitor from the blood. So we had to have something that was several log orders more sensitive than existing technology. So we're going to get a publication out of that. I'm sure there'll be some case study published to say, look, this approach worked in this way. And just keep in mind, it's work in progress, right? But to the point that Hugh made earlier, what I think long-term is going to get published out of this is a completely new type of scientific publication. It's not to say this drug has this effect on a patient with this mutation. It's basically say this approach of basically picking combinations of drugs based on mutations and then iterating and changing that if things are going in the wrong direction. In 50% of patients, we were able to basically change the patient from a growing tumor burden to stable or declining tumor burden, right? So the algorithm, like you were saying earlier, that approach is what's going to ultimately get approved and valued, not the particular, not the particular drug and the particular mutation, right? And that's the only way you're ever going to get enough sample data, because if you look at those individual combinations, there's never enough patients out there to have enough people in the population to be able to publish something.
So if anyone's interested in looking that up, look up Bayesian adaptive platforms. It's a very different way to studying things than randomized controlled trials. I think I'm just going to wrap up now, because I think we're just about at the end of... we've got one question here. I'll repeat the question. I think we need a microphone, actually. Sorry about this, because this is a long question. Yeah, but we're just going to be in silence for a bit. So please keep the question short.
Yeah. Okay. I completely agree with the idea of individualized tracking and treatment. The question twofold. One, for someone who's got the means, maybe 10,000 or 40,000 to access that, that is made possible. For the rest of the population, no. And whenever it comes to upscaling, it is always the big boys that have the capacity to do that. It's not the startups who have made the invention. So the question is, how do you expand once you approve the method? How do you expand it to millions of people who need the treatment? That's it.
Okay.
It becomes...
I will let Jo answer that, Joanna, and then we are going to wrap up.
Okay, so the question is really, how do you generalize these observations? And I think that is...
Scale up for the treatment of...
So we have to learn from the data, and we have to learn from data that people like Stefan are depositing to ask, are there generalized principles that we can use to develop therapies that are going to work for people without spending 40,000 up front? Exactly that. So we need that data. We need that data out there, and we need it at patient level so we can learn these.
And just remember, it cost $1 billion to get your gene sequenced 10 years ago. You can pee into a pot and do it almost for free in the next few years. So I have no doubt that the molecular understanding and tracking of these complex new therapies and algorithms will become as cheap as chips. Much cheaper than people like me.
And it shouldn't be that expensive. Keep that in mind. I think the costs are artificial.
Like
oh, yeah. It shouldn't be that expensive to sequence. It shouldn't be that expensive to develop these drugs. And we're going to be able to bring that cost down. It's not ideal to have this be something that only a few people have access to. But just like mobile phones were a luxury, and now you go to anywhere in Africa, everybody has a mobile phone, this will come down. The costs for all this are going to come down rapidly the more we do it.
Because some people had access, we're going to get there quicker.
Yep.
I have been fascinated by this panel myself. I hope you have too. Please thank for me Hugh and Fiona and Joanna and Stefan, and I look forward to a much brighter future for incurable cancer with the power of AI and emerging technologies.